You know, we are right on the edge of this huge leap forward in medicine, and it's all being driven by that same mRNA technology you've heard so much about. But here's the thing. To really design these life-saving tools, we have to become fluent in the language of our own biology. In this explainer, we're going to see how a truly groundbreaking AI is finally learning to speak that language, and with a fluency we've never seen before.
So let's start with a simple question. It's almost a philosophical one. In English, you could say fast, quick, or rapid, and they all pretty much mean the same thing, right? But you know, instinctively, that in some sentences, one of those words just feels right. It has the right rhythm, the right tone. Well, it turns out the exact same thing is happening inside our own genetic code. And this right here, this shows a central puzzle in genetics. Just imagine you have two different recipes for the exact same cake. In this case, a protein. When you give these recipes to our cells, one might crank out that protein like crazy, super efficiently, but the other recipe barely works at all. You're supposed to get the same exact thing, but the outcome is wildly different. So what gives?
Well, the answer is this thing called synonymous codons. You see, our genetic code is written in these little three-letter words called codons, but the code has a lot of redundancy. For example, there are 6 totally different three-letter words that all tell the cell, hey, add the amino acid serine right here. Basically, they're synonyms.
And that leads us straight to what scientists have dubbed the synonym paradox. It's a really simple idea, but it's also been this profound problem, a major hurdle for anyone trying to design effective mRNA therapies.
And this is where it gets really important. The choice of which synonym you use matters a lot. It can change how long the mRNA sticks around, how fast the cell reads it, and ultimately, how much of that critical protein gets made. For something like a vaccine or a gene therapy, this is everything. I mean, it is literally the difference between success and failure.
For years, we've been trying to teach AI how to write the best possible mRNA sequences. But the models, they were basically tone-deaf. They looked at all these synonyms and just saw them as interchangeable parts, completely missing the subtle, underlying grammar that our cells have been perfecting for billions of years. They just couldn't solve the paradox. Until now.
So how on earth did researchers crack this? Well, they had a brilliant insight. They stopped treating codons like just a boring list of words, and instead they started thinking about their relationships geometrically. And this led to a brand new model called Equi-mRNA, which is built around this beautifully elegant idea, the codon wheel.
Okay, so here's the core idea. The AI doesn't just see a random jumble of synonyms anymore. Instead, it groups them together and places them on a wheel, where every single synonym for a particular amino acid is an evenly spaced point right on the circumference.
This is the real game changer. The old models thought swapping a synonym was like pulling a completely different item off a shelf. But Equi-mRNA gets it. It understands that swapping one for another isn't some random jump, it's a predictable, smooth rotation around that wheel.
Now, the fancy math term for this is SO2 equivariance. But all that really means is that the AI has a built-in understanding of symmetry. It just knows that if you rotate a codon on the wheel, its properties should also change in a consistent, predictable way. It's not tone-deaf anymore. It can finally hear the harmony in the code.
So the big question is, does this new way of thinking actually work? Well, the results are in, and honestly, they're nothing short of remarkable. Equi-RNA isn't just a little bit better. It's smarter, faster, and just fundamentally more in tune with how biology actually works.
First up, when you ask it to predict the properties of an mRNA sequence, like how much protein it's going to make, Equi-mRNA is up to 10% more accurate than the old models. That's a pretty significant jump in precision.
But this is where it gets really impressive. When you ask the AI to actually generate new mRNA sequences, the code it writes is up to 4 times more realistic. And what that means is it looks a whole lot more like the kind of sequences we find in nature, way more than what the older models could ever dream of producing.
And crucially, those sequences that it generates are up to 28% better at actually preserving the functional properties of the protein they're supposed to create. So it's not just writing code that looks right, it's writing code that works right.
And what's maybe most surprising of all is just how efficient it is. You'd think a smarter model would have to be bigger and more complex, right? But Equi-mRNA hits this state-of-the-art performance with just 15 million parameters. That is less than a third of the size of its main competitors. It's just doing more with less. Okay, this graph is my absolute favorite part. The researchers added something called temperature. You can think of it as creative pressure or just a bit of randomness. Now, for the old model, shown in black, the quality just plummets.
But look at Equi-mRNA in color. Its performance actually improves. Its understanding of the structure is so solid that a little bit of chaos actually helps it find even better solutions. That's amazing.
So what does all of this mean for you and me? I mean, this isn't just some cool academic exercise. This breakthrough has direct, powerful implications for how we design the medicines of the future.
Suddenly, the whole process gets a lot more streamlined. Scientists can pinpoint a protein they need, maybe one that fights cancer or fixes a genetic disorder. Then they can use Equi-mRNA to write the absolute best, most efficient, most stable mRNA recipe to produce it. And the result? A much faster path to better medicines for all of us.
The researchers who built this put it perfectly. They said this isn't just another small improvement. It's a new biologically principled paradigm, a whole new way of thinking about mRNA that's actually based on the deep, symmetrical rules of biology itself.
You know, all of this leaves us with some pretty profound questions. For the very first time, we're moving beyond just reading the genetic code to truly writing it. We're learning its grammar, its nuance, even its poetry. And that brings us to one final, powerful thought. Now that we can speak the language of life this fluently, what are the first things we should choose to say?